Okay, so we left off in our notes about how yeast communicate through cell signaling. So we're on this section called local and long distance signaling. So cells in a multicellular organism communicate by, we're going to write chemical messengers. Animal and plant cells have what we call cell junctions, that's the J, that directly connect the cytoplasm of adjacent cells. What in the world does that mean? All right, so in animal cells, they have what we call gap junctions. And in plant cells, these connections are called plasmodesmata. And I'll come back to this here in just a second because I want to show you this picture here of this. So this picture here shows you the cell junctions. So here's your animal cell and here's your plant cell. Notice here that adjacent animal cells, you see the membrane here is perforated, there's holes in it, so that means that cytoplasm and any molecules that are in it can diffuse from cell to cell. So that aids in cell communication as well. Like there's a little circle here and a little triangle here showing you that cells can communicate that way. And then the chemical messengers here between plant cells, we have holes in the cell wall called the plasmodesmata, and the same thing is going on. So these molecules can diffuse back and forth, which aids in cell communication. So that's your cell junctions. The second type, it says in local signaling, animal cells may communicate by direct contact. So in this case here, they're directly in contact with one another, and can communicate. And I'm going to put some examples here. Our immune response in our immune system exhibits this, as well as early on in our development. So what becomes a head and a tail, and so on. Embryonic development communicate that way. So if we look at that second picture down over here, that's cell-to-cell recognition. So this is, see how this molecule right here is coming out of this cell. It's in the cell membrane, so that's a protein. And notice then that this cell has a complementary protein that fits that shape here. All of these here, that knob shape can fit into here, and when that happens, when this receptor here, this receptor binds into that, it causes a response to happen inside the cell. So kind of like what we talked about with the signal transduction pathway, that triggers changes to happen inside the cell so that the cell elicits a response that it wouldn't have elicited if these two weren't adjacent to one another and that connection weren't made. So in many other cases, animal cells communicate using local regulators, and the key there is local. Local means close by, right? So messenger molecules that travel only short distances. So they don't have to move very far. So the cells that are communicating are close together. One releases a molecule, a regulator, goes in short distances, and can communicate. That's what this down here is showing you here. So in local signaling, we have paracrine signaling and synaptic signaling here. Paracrine signaling, so here you have your secreting cell that's releasing your regulator molecules, the molecules that are going to communicate. So it diffuses as they're let out. Notice that they're being let out but through exocytosis, that they're in vesicles and then fuse with the membrane and then enter into the extracellular fluid. And so then they're just bouncing around, they diffuse around and get to the target cell, and they can bind to the receptor protein on the target cell and elicit response in the target cell. So these don't have to go very far because the target cell and the secreting cell are in close proximity to one another. Same type of thing goes on in our nervous system. Our neurons that work with our nervous system release a chemical called a neurotransmitter. It's just a special kind of chemical signaling. And so one neuron, this is your electrical impulse coming down your neuron. It gets to the end of the neuron, and how does one neuron communicate with another? It releases neurotransmitters. So here's that exocytosis here, releasing the neurotransmitters, and then the neurotransmitters bind to the next cell, which then triggers a response in the next cell. And that's how neurons communicate with one another. These are all local because the cells, the secreting cell and the target cell, are in close proximity to one another. Now, that's not always the case. In long distance signaling, plants and animals use chemicals called hormones. And so this is long distance signaling. So if we look here, this is how hormones work. That's called your endocrine system. So this is the endocrine cell, and here are your hormones in these vesicles, and the vesicles fuse with the membrane. And what we see here is it comes out through exocytosis and enters into the bloodstream. So this is in one part of your body. So they're showing you here, all right, that's in this part of the body, so upper body here. And it's coming down here and going to communicate with a part in the lower body. So that's far away for a cell. And so how does the hormone that's released from a cell up here get to a cell down here? And that's through your bloodstream. So these hormones flow through your bloodstream from upper to lower body, and then leave the bloodstream and bind to the appropriate receptor proteins within the membrane. So those are those receptors there, and the molecule binds to it and elicits a response in the target cell. And so that causes a transduction pathway that will elicit a response. And so these hormones don't affect any of the other cells because the other cells in your body don't have the right receptor proteins. So they have to have the right receptor proteins in the membranes of the cell in order to have the hormone affect it. So the hormone doesn't affect all cells. And so therefore the response happens just in your target cell, and therefore is specific to a cell type. And so that's long distance signaling because it has to move a long distance. All right, so let's look at then more specifically about what happens in this signaling process. So when the signal gets to the target cell, what actually happens to it? So this man named Sutherland discovered how the hormone epinephrine acts on cells. So he studied this hormone, and what he figured out is that there are three major steps for cell signaling. They're called reception, transduction, and response. And so what this picture is showing you is those three steps. So here we have, this right here is our plasma membrane, right? And our plasma membrane, remember, is a lipid bilayer, so that's what that is. And then we have proteins embedded within the membrane, and so this happens to be the receptor protein. So here you have your signal molecule. So an example would be like the hormone epinephrine. And the signal molecule, if this is a hormone, comes from another part of the body through the bloodstream and gets to your target cell, and that signal molecule can bind to that receptor because it has a complementary shape to that signal molecule. And so then they bind to that. That binding is called reception. And so then what happens, notice the signal molecule isn't entering into the cell, but yet you're going to get a change to happen inside the cell. And so you get that transduction pathway that I was talking about yesterday. Relay molecules in a signal transduction pathway. So this molecule can cause changes, reactions to occur in the cell. Sometimes this is an enzyme that causes certain reactions to happen, and so then you get this pathway of reactions that occur, and at the end you get the activation of a cellular response, and that's your response. So we have our reception, our transduction pathway, and our response at the end. So what I'd like to do is just talk a minute about the reception here. So 11.2 is all about reception. A signal molecule binds to a receptor protein, causing it to change shape. So we're going to talk about this, and I'm going to give you a couple examples of this happening. So the binding between a signal molecule, and now I'm going to introduce a new word here, a ligand. A ligand is another word for a signal molecule. And receptor is highly specific. So meaning it's kind of like an enzyme and its substrate. So the shape here is very specific of the protein is very specific to the signal molecule. So what happens when that signal molecule binds to that receptor is a conformational change. What does conformation mean? Again, it means shape. So we're really talking about, I'll put in parentheses, a shape change. That's a C. And a receptor is often the initial transduction of the signal. So what happens is when that signal molecule binds, you get a shape change in the receptor, and that's going to trigger the transduction pathway, which is the next step. Most signal receptors are plasma membrane proteins. So that's what we've been talking about here. So they're proteins. So now, the receptor, notice here it says intracellular receptors. So intracellular, the receptor can be either as part of the membrane or inside the cell. So some receptor proteins are intracellular, found in the cytosol or nucleus of target cells. So not all of them are in the membrane. Some of them can be inside the cell. So what kinds of if the receptor protein is inside the cell, so if I draw here the receptor protein inside the cell instead of part of the membrane, then what has to be true about the signal molecule? The signal molecule, if this is the case, the signal molecule needs to be able to get through the membrane. It has to bypass through the membrane. So what kinds of molecules can get through the lipid bilayer? See if you can answer that. If you remember, the lipid bilayer is mostly the hydrophobic tails, and so therefore the molecule has to be small or hydrophobic to get through that phospholipid tails. And so those small hydrophobic molecules, these are, let's put that in here, small and hydrophobic. To get through the lipid bilayer and go inside the cell to reach its receptor to cause a change to happen. So certain hormones are steroids. Steroids are a type of lipid, if you recall, and so some steroid hormones actually get through here. So the examples of hydrophobic messengers are steroid and thyroid hormones of animals. So steroids can actually get through, and so they can they still cause the transduction and the response, but the receptor is just inside the cell. So then what happens often with steroid hormones when the receptor is inside the cell is that when the signal molecule binds to the receptor, that it becomes an activated hormone receptor complex. That's the receptor, and this is the hormone, can act as a transcription factor. And what does that mean? That means it turns on specific genes. So remember, we talked about earlier what genes are. Genes are segments of DNA. So you have, here's gene A, part of the DNA, and here's gene B. Whoops, I'm kind of off the thing. Sorry, guys. I'm gene B. And so So what do genes do? They code for making polypeptides. So gene A codes for making polypeptide A, and gene B codes for making polypeptide B, and so on. Well, what happens is they found that sometimes these genes are turned off and they don't work unless they're turned on by a signal molecule. All right, and this activated hormone receptor complex acts as a way to turn these genes on. So how does that happen? So what happens when the genes are turned on, the polypeptide is made, and once the polypeptide is made, that polypeptide does something. It has some kind of job, and that's what elicits the response. So when you look here at this next picture, that's what we see here. So we have hormone, testosterone. Testosterone is a steroid hormone. So what did we just learn about steroids? We know that they're a lipid, so they can actually get through the plasma membrane. So this is the plasma membrane right here. It's the thickness right here. And so the steroid hormone can go right through the plasma membrane, and here's its receptor protein inside the cell. And so the steroid, the testosterone, binds to the receptor. So here's your hormone receptor complex. Notice here that they can get through. This is the nucleus right here. So here's the nucleus. But notice here's, remember the nucleus has a double membrane. So here's the double membrane of the nucleus right here. And then notice here that there are openings right here and right here. There's openings. Those are the nuclear pores. And the hormone receptor complex can actually go into the nucleus, bind to a specific part of the DNA, and that binding of it, that when it binds to the specific gene, is what turns the gene on. So then what it does is when it turns the gene on, it makes an mRNA, which then leaves the nucleus and goes to, these guys here are your ribosomes. And the mRNA, this is your mRNA, is read and is used to make your protein. So each of these little balls are amino acids, and so you're getting your chain of amino acid. And so then the protein will then give a specific response. And so that's how a lot of steroid hormones work, and they usually turn on genes when that happens. So this gene, whatever it is, wouldn't have been turned on if testosterone hadn't come in and bound to the receptor protein. If there was no hormone, then the receptor proteins would just be hanging out in the cell doing nothing. All right, and that gene would always be turned off. But when testosterone enters, then it's able to turn that gene on. All right, and so that is intracellular receptors, when the receptors are inside the cell. Now, we've talked already a lot about how the receptors are in the plasma membrane. So what kinds of things have the receptors in the plasma membrane, like this one right here, is signal molecules that don't readily diffuse through the lipid bilayer. So most, we put here, most water-soluble, so that's polar, hydrophilic things are not going to be able to get through the membrane. And so most water-soluble signal molecules bind to specific sites on receptor proteins in the plasma membrane. So they cannot get through. And so I'm going to give you a few different examples of this happening. So there's a G protein-linked receptors, receptor tyrosine kinases, and ion channel receptors. So we're going to look at the first one first. A G protein-linked receptor is a plasma membrane receptor that works with the help of what we call a G protein. The G protein acts as an on-off switch. If GDP is bound to the G protein is inactive. What in the heck is GDP? They're kind of like ATP and ADP. ATP has adenine. GTP and GDP has the base guanine. Adenine and guanine are two of the bases found in DNA. And so we have something called GTP that can break down into GDP and a phosphate, kind of like how ATP can break down into ADP in a phosphate, and this reaction is reversible just like ATP is. So you have this GDP bound to the G protein, and it's inactive. So let's turn the page here and look at this picture. So this picture here is just showing you, here's your lipid bilayer, that when we talk about a protein embedded in a membrane like this here, remember proteins are chains of amino acids that have the alpha helix and the beta pleated sheets and so on. So this is just pulling back, way back from when we talked about the structure of proteins. So here's your signal binding site. So this is where the ligand, the chemical signal, will bind. And then this says right here, segment that interacts with G proteins. And so let's look at that here in this picture. So what do we have here? Well, this picture is in your book too, so you can refer to it in your book as well. And it gives explanations underneath here as well if you'd like to look at that as a resource. So here's your G protein linked receptor. Here is that signal binding site that we just talked about here. So that's the signal binding site. Now notice here in the plasma membrane you also have a G protein that's inactive. And we just wrote that this G protein is inactive when GDP is attached to it. So look at here, GDP is attached to it, so it is in the inactive state, it's not doing anything. And then look at here, we have a third type of protein in the membrane. So we've got 3 different kinds of proteins, and this is the enzyme. This is an enzyme that's going to speed up a reaction, but right now this enzyme is not working, and it usually doesn't work because it's not in the right shape, right? So right now it's inactive. So we need to activate this enzyme, and that's what's going to start the response, the transduction pathway and the response inside the cell. And so what happens? So you have a ligand is your signal molecule. It binds to that special spot in the G protein linked receptor. This is now an activated receptor. What happens next is that triggers GTP. So GDP, remember GDP was attached to it. The GDP becomes detached. So this detaches. And what comes in right here is GTP attaches. And what is it attaching to? The G protein. All right, so that's what we have here. So then once GTP attaches, what does that trigger to happen? Look at here, the GTP diffuses through the membrane and comes over here and binds to the enzyme. And what does it do to the enzyme? It activates it and activates it. By how? Look at here. It has changed its shape by changing its shape. And so that's what we have. We have this activated enzyme now. Notice that once that happens, the signal molecule detaches and this becomes inactive again. But what we've gotten is our response. We've got this activated enzyme that is now going to eventually elicit a cellular response. And that activated enzyme wouldn't be activated without that signal molecule. So that signal molecule indirectly activates an enzyme, which then is going to elicit a cellular response. Once that happens, the G protein has done its job. It loses a phosphate and becomes GDP attached to it, and now the G protein is inactive again. And so this is your G protein. Let's label that. And it's inactive, and it sits there until another ligand comes and binds to, another ligand binds here. Another ligand binds to the receptor. And remember then after this detaches, then the enzyme is inactive again. And it won't become activated again until another signal molecule comes, another ligand comes. And so that's one example of a receptor protein in the membrane and how a ligand comes and binds to it, activates an enzyme that triggers a transduction pathway which will lead to a cellular response. So that's example 1. Example 2 is with receptor tyrosine kinases. These guys are membrane receptors that attach phosphates to tyrosines. A kinase is, notice here it ends in ase, right? Kinases are enzymes that attach phosphates to something, all right? So that's what those are. So a receptor tyrosine kinase can trigger, they can trigger multiple signal transduction pathways at once. So they can cause several different cellular responses. And unlike the one that we just talked about here, one signal molecule binds, activates one enzyme, triggers one cellular response. This can do multiple ones at once. So let's look at how it can do multiple ones at once. So here we have, let's look at what we have here. This is the receptor tyrosine proteins. These are inactive. And so there are two parts here. They're eventually, you're going to see here, come together to become activated. But so right now they're inactive. There's two parts to it. And so these are the tyrosines, parts of it. And these are your signal binding sites. So notice here there's two signal binding sites in each one of these parts. And so in comes your signal molecule. Your signal molecules, notice here it takes two. Your signal molecules bind to that receptor site. And once they do, what does it happen? The two parts come together. All right, and so that's what we have here. So then what happens? So the next thing that happens is that this is now what we call an activated tyrosine kinase. And so because the signal molecules are bound together and the whole thing came together, what these tyrosine kinases do, these act as enzymes. These little, right here, tyrosine kinases, they act as enzymes, and they're going to phosphorylate. And so they phosphorylate themselves. So this fully activated receptor tyrosine kinase, it's phosphorylated. How do we get the phosphates to phosphorylate them? From ATP. So notice that there are 6 tyrosines and there are 6 ATPs, and breaking down into 6 ADPs, and 6 phosphates are added. Now we have a fully activated receptor tyrosine kinase receptor molecule. So what do these fully phosphorylated tyrosines do? What they do is they activate what we call relay proteins, which triggers your signal transduction pathway. So this molecule becomes activated, which then activates another molecule, which activates another molecule, and so on. And so this right here, these arrows here, represents a signal transduction pathway. And so one thing turns into another, and eventually you get your cellular response. But notice here that we have different relay proteins. So this one right here, look at the shape of this one, and then look at the shape of this one. They're different shapes. They're different relay proteins. And so this relay protein elicits a signal transduction pathway to give you one cellular response inside the cell. And then this one binds to it and gets activated and triggers another signal transduction pathway that gives you a totally different cellular response. And so that's what that means when we wrote over here that a receptor tyrosine kinase can trigger multiple signal transduction pathways at once. And so you have all different types, and you can see here you can do up to 6 different cellular responses. This picture is only showing you one. All right, and so that's your receptor tyrosine kinase, all right, eliciting multiple responses from your ligands. But still, an example of a receptor in the membrane. And then lastly, we have our ion channel receptor that acts as a gate when the receptor changes shape. When a signal molecule binds as a ligand, the gate allows specific ions such as sodium or calcium ions through a channel in the receptor. And so let's look at this picture here, and this will be the last picture we'll do for the day. So here again is your plasma membrane right here, and here is your gate. So this part right here, I'm going to highlight right here. Notice here the gate is closed. This is the part right here. There's a channel through here, but the gate is closed. That's not going to let the molecules through. So you have these ions in high concentration of ions outside, low concentration of ions inside. And so ions will want to go in from high to low concentration, and it has this ligand-gated ion channel, but the ions can't get through because the gate is shut. So the signal molecule binds into the spot that it fits into the receptor, and so when it binds, if you notice here, what happens? That gate now is open, and so now these ions can diffuse in, this is diffusion, from high to low concentration into the cell, and then what happens is when that signal molecule detaches, so the signal molecule detaches, notice that the gate closes again. And so the gate only opens and closes in response to a signal molecule. An example, this happens in our neurons with neurotransmitters. So when a neurotransmitter is released from one neuron and binds to the next neuron, it binds to it and causes the gates to open and ions to come in, which then triggers the next cell to send a message. All right, and so that's how those work. So that is the examples of reception. So I'll leave that here for today, and tomorrow we'll look at the transduction part. So what happens in the signal transduction pathway. All right, have a good night.